Hello everyone. Today I am going to talk about the drug target coronavirus, and I will find out the active molecule from the database for this COVID-19 and try to give you guys some heads up how I can do the lead optimization. So let's go to CMD, type Jupyter Notebook, so the home page of Jupyter Notebook opens. So go to the new page and give this page name, so COVID-19. So now in order to work with medicinal chemistry, I have to upload number of library. So just I'm going to copy and paste these libraries from the notepad, copy all the libraries and paste it in the cell. So here you can see the common library are NumPy, Pandas, Matplotlib, and some medicinal chemistry libraries are need to import RDKit, and from RDKit import all of the chem libraries. And I'm going to open the open source Campbell web page. So import the new client, and for bioinformatics, for 3D structure, I can import also some libraries as well. So first get the target from the database. Target new client dot target dot search. The coronavirus target, and make this target to table format. So from this table you can see the SARS coronavirus 3C like protein is. I'm going to use this single protein, and then index is 4. So get this target out from the table. Campbell ID I'm going to use the 4, index 4. If I run it, you can see this Campbell ID 3927, and this ID is 3927. From using this target 3927, I'm going to pull all the active data from this database. So write down the code. New client activity dot filter and then target Campbell ID equals to target, and then also filter IC50, filter type equals to IC50. Also make this variable to table format. Now I can run this and see the shape of the table. So it has a 105 rows and 43 columns. So see the columns of the table. As you can see, all the column is showing over here. So I'm going to use only the column I need to work with. So I will create a data frame. I'm going to take the molecular Campbell ID, the canonical smile, the type, and also I'm going to use the standard value. And check these few rows. Here, as you can see, the IDs and the one-dimensional structure. I filter only the types, so only I can see IC50. There are so many different types as well: KI, EC50, percentage, activities. So different type of you can see also, but I just did it only the IC50 and their nominal level activities. So just first clean up this table if there is any non-value. So now I am going to add, as you know that the medicinal chemist, they always like to see the structure of the molecule. So I am going to introduce the molecular structure into this table. So pandas tools dot add column molecule columns molecular column to frame. So this canonical smile to make it to the molecule. And see this view head of the so what is the issue here? Canonical smile or canonical smiles has to be S here. So run it. As you can see, here you can see all the molecule of the from it coming from the canonical smile. We can convert it to all the molecules. So now as you know that one is octane water partition coefficient, the log P, one of the very important feature. It shows a molecule will behave inside the body, that the absorption, distribution, metabolism and excretion depend on log P value. So let's introduce the log P also in this table. descriptor dot molecular log p, then appended log p. So run this. Name log is not defined. Okay, log p. Oh, here is the p missing. So now this mlog p, this create as a series, so I have to convert it to the tables. So write down the code is df1 equals to pd dot data frame log p columns equals to, give the name of the column, sorry, log p. If we run this, now you can see the log p only giving all the log p. So I have to concatenate these log p tables with this table. So this is df and this is df1. So if I join these two, df2 equals to pd concat df1, sorry, df1 axis equals to 1. For row is 0, for column is 1. So now if we run this, head of few column. So now we can see the molecular structure and log p. You can see the log p here. So now we have a molecule and we have a molecular activities. We also have a log p. So now we wanted to see the most active compound from this table. So in order to do that, first I have to see all the data type for these tables. So df2 dot data type, you can see here most of the data types is objective. This is integer, I mean this is string. So the stand value we can is all the numeric here. So in order to work with this, I have to convert this object to numeric or float. So let's convert it. Dot standard value dot as type float. So now we converted to float. So if I run back this one, you can see this is standard value converted to float. So now I am going to get most active compound from this table. So this is similar, so I just copy and paste this code and convert it here. So this is a boolean, so it's need to double df2, sorry, df2. So now if I run this, you will see the most active compound from this table. So this is the most active compound in this database. A molecular weight for this compound is less than 500, so it is drug like molecule. Log P only 2.6 is a little high. Usually intestine absorption happen between 1.4 to 1.8 and CNS absorption too. So if we consider this is a lead molecule, then this molecule need to be optimized. As you can see, this molecule can be divided into three parts. The left part A is 5-member ring furan, the middle part B is linkage, the ester, and the right part C is bromopyridine. To optimize, keep 2 parts constant and modify 1 part at a time, and try to improve the activity and the log P. So now if we just wanted to see the 3D structure for this molecule, and then get the protein and how we can do the docking with the active site. So let's see the 3D structure for this active molecule. By 3D dot view query. Here this molecular chemical ID is this. If we just copy this ID and go to and write chemical ID pubchem, here you can copy and paste this, and the first molecule you can see the same molecule is this from the pubchem. So we can copy this ID and paste in over here. And put this set style stick spectrum. Run this. Now we can run this, and you can see the 3D structure for this active molecule. And also if you wanted to see the coronavirus the protein, just it's very similar. Copy this code and change this ID. This is from the protein database, the PDB. And if I run this, you see that all this protein is structured with a stick. And if you wanted to see the cartoon, you can see the cartoon. The next step would be how we can take this active molecule and docking with this protein, and that we will do at the next time. Thank you for watching. Thanks.